Dear guests, dear colleagues, I would like to introduce today a distinguished speaker. But first, I would like to say that this talk today is organized within the project Horizon 2020 Twinning, titled MICOBION, and that stands for abbreviation microbial communities in biomedical and environmental areas and system biology. This project is coordinated by Faculty of Science, Charles University, under the leadership of Professor Matthijnssens. Because in the time of coronavirus pandemics, we can't meet personally, and the situation lasts over a year, we decided to organize lectures planned within this program online. We would like to invite one speaker every month from January till June, and provide access for broad scientific audience to these events. We hope that we will not face any technical difficulties today, but on contrary to our government, I claim that even this might happen because today is the first run of these online lectures from BioCEF. Now I would like to introduce the speaker, as I mentioned. It's Professor Jelle Matthijnssens. He studied on the Faculty of Applied Biological Sciences on the University in Leuven and obtained his master's degree diploma in 2003. His doctoral thesis he finished in 2008 in the Laboratory of Clinical and Epidemiological Virology at the Rega Institute for Medical Research, Catholic University in Leuven. His thesis subject was genetic diversity and classification of human and animal rotaviruses. And in 2020, he qualified as an associate professor in Catholic University in Leuven. He joined several research projects abroad, for example, on Yale University, New Haven, Connecticut, USA, at the National Center for Agroindustry Investigation in Buenos Aires, Argentina, in the National Institute for Veterinary Research in Hanoi, Vietnam, and in the Federal Institute for Risk Assessment, Berlin, Germany. From 2014, he is a chairman of the study group Reoviridae in the International Committee on Taxonomy of Viruses. He's teaching several courses at the University of Leuven as well as abroad. He also was an organizer of several meetings in Belgium and abroad. Jelle is an author and co-author of 160 articles in the peer-reviewed journal, and now he is a principal investigator of the Laboratory of Viral Metagenomics in Catholic University in Leuven. We met with Jelle on the projects financed by CELSA, which stands for Central Europe Leuven Strategies Alliance between Catholic University of Leuven and 6 other universities from Hungary, Slovenia, Czech Republic. The CELSA cooperation agreement was signed in 2016, and the first call for joint project was in 2017, and other projects called Metagenomics analysis of the virome of the honeybee in the Czech Republic obtained financial support. So I would like to invite Jelle. The stage is yours, Jelle, and we are looking forward to your lecture today. Thank you.
All right. Thank you very much for this very kind introduction. Just a double check. Can you hear me fine? Yes.
I think you, yeah.
Can you hear me?
Yes, we can hear you.
All right, perfect. Yeah, so Ruth, thank you very much for this very kind introduction. And as you already mentioned, I started my PhD about almost 18 years ago on rotaviruses. And at that time, we didn't have next generation sequencing yet. So we had to do everything with classical Sanger sequencing. And when I wanted to sequence a complete rotavirus genome, it could take me many weeks up to months doing that. And all these things changed quite drastically in the last number of years, almost a decade, with the introduction of next generation sequencing, which really changed the field. So we also jumped on this when we studied viromes. And in order to do that, first, a former PhD student in our lab, Nadia, developed a method, a protocol to purify viral particles from biological samples which then could be used for next generation sequencing, which allowed us to study entire viromes, looking at all the viruses present in a particular sample. And that paper was published in 2015 in Scientific Reports and is schematically shown on the right here. So what we actually did was just we used a number of physical methods to purify viral-like particles from biological samples. And I'm not going to go through the entire protocol, but in a nutshell, you purify viral-like particles, you extract all their nucleic acids, do random amplification, and use deep sequencing, in our case Illumina, to look at all the viruses present in a sample. And initially this protocol was developed mainly for using on stool samples, but as you will see in the presentation I will give, we are now using it in a number of different biological samples as well. So actually in the next 45 minutes, I want to tell you about 3 projects. And these are actually 3 projects that were performed by 3 PhD students in our lab that defended their thesis in the last year. And these projects are about the virome of mosquitoes, honeybees, as well as the gut of infants. So I actually shamelessly recycled some of the slides they generated for this presentation today. So I would like to start first with the mosquito virome, and that was done by Shenyan Shi, a student from China, and she's currently back to China doing a postdoc there. So the hypothesis of her PhD thesis was that insect-specific viruses might be able to influence the vector competence of mosquitoes for important human arboviruses or arthropod-borne viruses. We all know viruses such as dengue virus, Zika virus, yellow fever, etc., which are carried by mosquitoes. But some of these viruses can only be carried by particular sets of mosquitoes, and even inside a particular species of mosquitoes, it was not such that every mosquito was able to transmit the virus to a similar extent. So in order to better understand this, we hypothesized that insect-specific viruses, so viruses that infect the mosquito, but that cannot infect humans or animals, might influence this. And to start that, we needed to have a good idea about the virome present in mosquitoes. So in order to first start to study this, we collaborated with researchers from Guadeloupe, a Caribbean island, who provided us with samples. And initially they provided us with samples from 2016, which were collected in Les Abîmes, which is one of the 2 parts of the island. And we pooled them according to the species, Aedes aegypti and Culex quinquefasciatus, and also based on their gender. So we pooled those mosquitoes and we sequenced them. We found a range of novel viruses. But then next question was, are we able to perform viral metagenomics on individual samples instead of on pools of samples? So what Shenyan did next was we received a new set of samples from Guadeloupe, and she divided these samples, of course, from the 2 different islands, from the different species, and then male, female. And she, for each of these 6 categories, she made 5 pools of an individual mosquito, and 1 pool of 5 mosquitoes to compare and to see if it was feasible to do viral metagenomics on single mosquitoes. And actually, it turned out that it worked quite well. And all right, let's see, can switch to the next slide. All right. So what you can see here is just a rough overview, first separating the data by eukaryotic virus and bacteriophages. So what you see on the left panel is the data from the Aedes aegypti samples or pools. So the samples here, the columns with a red dot, these are pools, and the other ones are individual samples. There's one sample here with the asterisk, and I will come back to that one in a second. So this for Aedes and for Culex. And as you can see, for Aedes, the majority of this virome was dominated by eukaryotic viruses, and only few bacteriophage reads or contexts were identified, which was a bit different from Culex, where we actually found also quite a high number of bacteriophage contexts. And I will come back to those bacteriophages also shortly at the end of this part of the presentation. So the majority are still eukaryotic viruses. So when we perform an NMDS analysis, which is similar to a principal and component analysis, where we compare the virome profiles of these different samples, we could see clearly that there is a separation between the Aedes viromes and the Culex viromes, with one exception here, up here, which was an outlier, which I will come back to also later. So this one seemed to have quite a distinct virome from the other mosquitoes, the other Aedes mosquitoes. So then when we looked in more detail about which viruses we found, I want to explain you this slide. It's quite complicated, but I will guide you through. So first, all the contexts that Shen-Yan identified, we annotated and we compared them to databases. And actually we found that the closest hit in database was this list of different eukaryotic viruses. And of course, in some cases, the hit we find is quite close to something in databases, and in other cases, it's quite divergent. And that is kind of shown in this with these blue boxes here. If it's dark blue, for example, this guy here, it means that the sequence we found was very similar to a virus already known and present in databases. Whereas for other ones, for example, this one here, the skull, this river bee virus, is light blue. So the similarity between what we found and sequence in database was below 40% on the amino acid level. So highly divergent viruses. So you have to be a bit careful with interpreting this. This is the closest relative, but it doesn't really mean that that's the one we found, but at least a close relative. What you see on the bottom here, these are all, again, all the samples, either the pools with a red dot or the individual samples. And here, the one outlier I already discussed before. And now you can see probably why that was an outlier, because we find a number of viruses present only in that sample, which were not present in any of the other samples. So what you can see also here is that on top here, all the Aedes aegypti samples clustered nicely together, and the Culex as well, showing that they had distinct virome profiles. What you can also see is that it seems that many of these viruses are not found in one or a few mosquitoes, but actually that we found reads of these viruses in many of these samples. And as you can see, some viruses were present mainly in one species, but a few of them are kind of shared between both species. So we then further want to look into these viruses. And of course, not all of these viruses that are mentioned here, we have complete genomes. But when Shen-Yan looked closer into that, she was able to identify about 11 complete genomes of insect-specific viruses belonging to multiple genera, families, and orders. And I've shown two phylogenetic trees on the right side of this slide, and I know you cannot read it, but that's not really that important. But what I wanted to show here is that, for example, this cluster in red and orange, these were all the viruses that we identified in the Culex mosquito, in the Aedes mosquitoes, and in red are the ones from 2017, and in orange, the ones from 2016. And what you can see is that nearly all these viruses are very similar. They are nearly identical, both in 2016 and 17. And actually, we see the same for Culex mosquitoes down here. So it suggests that all these mosquitoes that we analyzed carried very similar viruses, also across different years, which suggests to us that these viruses are actually vertically transmitted from one generation to the next. And of course, what I showed in the previous slide, all these data, this is of course relative data based on the NGS data, but we also wanted to see if we looked more in a quantitative way. And what Shen-Yan did was she selected 6 of the viruses we found. The 2 top ones here are viruses that were found both in the Aedes and in the Culex. And then she also selected 2 viruses that were specific to Aedes and 2 viruses that were specific to Culex mosquitoes. And for these, she developed a qPCR assay. And then we used an additional set of mosquitoes from 2017, actually 72 individual Aedes mosquitoes and 24 Culex mosquitoes. And all these mosquitoes were tested with the qPCR for all these 6 qPCRs to get a better quantitative idea about the viral load of these different viruses in individual mosquitoes. And the results of all these qPCRs are shown here. So what you can see, the 4 panels on the left, these are all from Culex. The 4 panels on the right are Aedes. So these 2 panels here are from the Culex specific virus. The 2 on the right are from the 2 Aedes specific viruses. And then the 4 in the middle are from the 2 viruses that were found in both species. So what you can already see from this is that it seems that in the Aedes aegypti mosquitoes that the viral loads seem to be quite a bit higher than they were in the Culex, which was in line with what our NGS data showed. What we also see is that, well, these horizontal red lines are kind of arbitrary cutoffs that we used, about 10 genome copies per mosquito. And if you use that cutoff, you see that the majority of the tested mosquitoes contain traces or high viral loads of the viruses we checked. So it seemed that nearly all of these mosquitoes had these particular insect-specific viruses. And as I mentioned, most likely they're transmitted vertically. So this suggests that there is some type of core eukaryotic virome present in those mosquitoes, which is quite interesting with respect to our initial hypothesis that these insect-specific viruses might have a role in the influence of the ability of these mosquitoes to transmit arboviruses. So this is a bit of a summary slide of the previous one, where per mosquito we indicated the viral load of the 4 viruses we tested for each mosquito. So you can see that for the individual mosquitoes, which are all shown here, for Aedes, that the viral load ranges from about 100 million viral particles to a couple hundred. And of course, this is only just for the 4 viruses we tested. So probably if you would test for more viruses, this number would be even higher. For Culex, this number was slightly lower, ranging again from a couple hundred to about 10 million reads. So what I didn't tell you in the beginning, of course, we perform our nativir protocol to purify viral particles from our biological samples. But this process is never perfect, so there's always some host nucleic acids that get through and that get sequenced. So in this case, we actually used this. So we used all the non-viral reads and we compared them to a number of marker genes. So these are genes that are present in nearly all cellular organisms. For example, the COX-1 gene, which is cytochrome C oxidase subunit, the DNA gyrase subunit B, and a recombinase A protein. So they're database of these proteins, and we used the reads we have to map against those to see if we could identify which organisms were also present in our samples. So what you can see in this figure is a heat map. And again, down here, you have all the different pools or the individual mosquitoes. And these are all the marker genes for which we found reads that we had mapping to these marker genes. So what do I want to highlight here? So first on top, you can see here, these are the marker genes from the mosquitoes itself. So we have Aedes marker genes, and you can see that all the Aedes samples really have a lot of reads mapping to these marker genes that made sense. And for Culex, we also see a lot of Culex reads mapping here. But there's one outlier here, and that was again that one strain that one mosquito that also shared an unusual virome profile, and it also didn't map here. So most likely this was just a completely different species that was misannotated as Aedes. Furthermore, these are 3 bacterial marker genes that we found, and these are bacteria that are usually found in the environment. And then interestingly, we also found a number of hits to marker genes from Wolbachia. And Wolbachia is a bacterial strain that lives usually as an endosymbiont, so it lives inside the eukaryotic cells from mosquitoes, and that is mainly found in Culex mosquito. And also here, in multiple of our samples and pools, we identified marker genes from these Wolbachia endosymbionts. So although this was not the goal, these additional reads that were generated could also be used to do these analyses. So that was an interesting side effect. And then very shortly, I also mentioned that there were quite a number of bacteriophage contexts that we identified. So what Shen-Yan did with those was also, she didn't go into a whole lot of detail there, but one of the things she did was to try to predict the hosts of these phages. And for most of them, we found actually Wolbachia as a predicted host based on CRISPR analysis and based on tRNAs, transfer RNA, which are often present in phage genomes. And actually in this slide, all the names that are in black, these are phage contexts. And the ones with a gray background were the ones that we could identify based on CRISPR analysis that they most likely infected Wolbachia. So what Shen-Yan did was she looked at the profiles of the abundance profiles of these phage contexts and compared those to the abundance of reads mapping to the marker genes I mentioned before. And this type of correlation analysis, she could look at which contexts were found in the same sample. So if two particular contexts were found always in the same samples, there's this very strong correlation, which is indicated by red dots, as you can see here. So there were some patterns that came out here, and a few I want to highlight is first this one here. So this cluster actually contained a number of phage contexts, as well as marker genes against these two bacteria that I mentioned before. So this strongly suggests that these phage contexts actually infect these bacterial strains. Another set of viruses was shown here, and this was also some phage contexts, as you can see here, and some of these could be classified as Cystoviridae. This is a family of virus with a segmented genome, so we have these 3 segments of this virus that we could identify, and these cluster closely together with Culex marker genes, so suggesting that these phages infect the bacterium present in Culex. We didn't find any bacterial marker genes there. Most likely it means that this particular bacteria did not have representative genes in the database we used. And then the majority of the phage contexts clustered closely or had a strong correlation with contexts with marker genes of Wolbachia as well as Culex, which also further suggests that all these contexts are parts of viruses that infect these Wolbachia. And to date, there's only a few bacteriophages reported from Wolbachia, but this analysis suggests that there is a lot of phages still to be discovered and to be analyzed, and that these also could have an influence on the mosquito health or on its ability to serve as a competent factor for arboviruses. So to summarize this part, we showed that it was feasible to really use an individual mosquito for viral metagenomics analysis. We also showed that both tested mosquito species, Aedes and Culex, show a striking difference in the abundance and the diversity of eukaryotic viruses and phages. We could identify near complete genomes of 11 novel viruses. Most of them, well, or nearly all of them were probably mosquito specific viruses that were vertically transmitted over generations. And it also seemed that both of these species had their own set of stable set of core eukaryotic virome, if you want to call it like that, both from multiple mosquitoes as well as over time. And we also showed that there's a plethora, a large number of different phage contexts present, which most likely infect Wolbachia. At this point, Shen-Yan is still finalizing some work about what we really do in vivo experiments, really trying to get a bit more into the details of how these insect-specific viruses might actually influence the ability of mosquitoes to transmit important arboviruses. But this is still work ongoing, so who knows, I might be able to present that on a future presentation. So the second part of my presentation is about honeybees, and this work was done by Wart. He finished also his PhD in my lab, and he's currently working in Freiburg as a bioinformatician. So Wart, the hypothesis of his thesis was that there is a large underexplored virome in honeybees, and that those might potentially be a contributing factor to winter loss, an observation that many hives of bees are lost over winter. And actually this study was initiated a couple of years ago, actually kind of by accident, because of a colleague from our institute that came to me and said, well, I have another friend who is a beekeeper and he has some problems that he loses quite a bit of his colonies over winter, and he would like you to check some bees of his for viruses. So I said, sure, why not? And he gave me some bees, and those were, I got them in an envelope and they were squeezed and dried out. So certainly not ideal samples to work with. But lo and behold, when we analyzed them, we found quite a number of novel viruses. So this triggered us to further look into that into a more systematic way. And when we went to look into Belgium, who works with honeybees, we came across Professor Dirk de Graaf from the University of Ghent. And he's kind of a world-renowned expert in honeybees and honeybee viruses. So we went to talk to him, and he had a very interesting collection of honeybees that actually were collected in the framework of EpiLobby, which was a large EU-funded study on honeybee colony mortality. And that was a study that ran over 2 seasons, 2012-13 and 13-14. And what they actually did, they had 17 countries in Europe that participated in this study. And in this study, people went to different places in their country and identified hives, and they collected honeybees at the late summer, early autumn of these different hives. And then they went back after winter in spring and went to check to all these different hives, which hives had survived winter and which had not. So now they knew we have bees from a strong colony and from weak colonies. So we had access to these data for our study. So what you can also see on this figure on the right is the different countries are colored according to the percentage of hives that were lost over winter. So as you can see, Belgium is here. We had one of the highest mortalities, up to almost 1/3 of all hives in that season did not survive winter. So that is quite a devastating effect for beekeepers, that they lose so many of their hives over winter. So we were lucky to have access of these samples that Wart could use during his PhD. Actually, we had samples available from the northern part of Belgium, the Flemish part. And what you can see here is that the viruses or the samples that we used were equally spread across the different provinces in Belgium. And here we see in red and black dots the hives where they were located in Belgium in the 2012 and 13 collection periods. So what Wart did was, from these available samples, he collected 300 colonies and pooled bees from these colonies per tree according to the health status, weak or strong colonies based on winter loss, and based on the year 2012-13. So we ended up with 100 pools of honeybees, which he subjected to the Metavir protocol. And as you can expect, he found a very large number of viruses, and the most frequently found were Varroa destructor virus and deformed wing virus, which are very well-known viruses in the honeybee virus world. But he also found some more unusual virus strains, which I want to highlight a few from. And the first one was actually an orthomyxo-like virus. This is a group of viruses also including influenza. And the virus he found, which is shown in orange here, was most closely related to togotoviruses. And togotoviruses are a group of viruses that are known to be arboviruses. These viruses are found in ticks, as well as in birds and mammals. So these are known arboviruses. So the left part is based on RDRP. Also, when you looked at the other gene segments, because the segmented genome, so these viruses have genomes usually between 6 and 8 segments. So we looked at multiple gene segments. It also was most closely related to these togotoviruses. And this particular virus we were quite interested in, and we also wanted to see if we could find further real evidence of this virus in our colony. So the colleagues from Ghent screened with a qPCR for this virus for other hives, and they were able to identify a fresh hive that was infected, and they immediately used those samples for electron microscopy, and they were able to identify viral-like particles which most likely belong to this orthomyxo-like virus. A second virus I shortly want to talk about is Leuven virus. This is a group of viruses that was first discovered in the United States a while ago, but in the meantime it had been found by Professor de Graaf in Belgium already, and actually we also found different variants circulating in Belgium, which are all shown in orange here, over this period of 2 seasons, so it seemed to be a quite common virus in Belgian populations. And then a third virus which caught our attention appeared to be some type of recombinant. So when we first looked at the RDRP, the RNA-dependent RNA polymerase, this virus, which is shown in orange here, was quite divergent from astroviruses and Viridae families, but very divergent. But when we also looked further at that genome, so this is the tree that I just showed, which was based on the RDRP, but when we also looked at the capsid, then this capsid clustered distantly from the Leuven virus, so completely unrelated viruses, suggesting that this might have been some ancient recombination. And of course, with NGS analysis, you also always have to be very careful with artificially generated chimera, but looking at the profile of the read mapping and as well as confirming by Sanger sequencing, this was a genuine virus, so it's probably really a true recombinant. But again, we know very little about this virus, so more research is needed on this virus. Then a third virus I shortly want to mention is a Tika-like virus. So this virus was quite divergent, it's shown in red here, from a known virus in databases. And actually these viruses I'm showing here, they have not been officially classified yet by the ICTV. But the strain we found was most closely related to Tika viruses. And these were viruses that were initially identified in Drosophila. Yeah, so this is what we call Tika-like virus, highly divergent from any known viruses. And then there are 3 more viruses I quickly want to tell you a little bit about. And the reason I want to tell you a little bit about this is because these were viruses we found quite frequently in multiple tested samples. So I quickly want to tell you a little bit about them. So the first one on the left here is Apis mellifera filamentous virus. These are viruses with large double-stranded DNA genomes, highly divergent from some other large DNA viruses, such as pox virus or baculoviruses. So these have been described before, but there is relatively little known about these viruses, but they have very large genomes. The second group of viruses here are we call Macular-Like Virus. They're again quite distinct from known viruses. So there are a few hits in GenBank that are closely related, but these also have not been classified at this moment. So little we know about it, but it's most closely related to known Macular-Like viruses. So we probably need to come up with another name, but for now, the time being, we call it Macular-Like virus. And then a third one that we found quite frequently in our data was actually a virus that was related to Partiti-like viruses. And Partiti viruses were traditionally mainly seen as viruses infecting fungi and sometimes plants, but actually recent metagenomic data have shown that actually these viruses are found also in large numbers of insect samples. So suggesting that this virus also might also infect insects, but again, further research needs to be done there. But as you can see here, there's one particular virus that we found in many of our samples. And again, we're not really sure if it really infects honeybees, but the fact that we found it in many of our honeybee samples spread all over Belgium suggests that it actually might infect honeybees as well. So the next thing War did was he developed also, similar way as Shenyan did, qPCR assays for some of the viruses that were frequently identified in Belgium to again have a more quantitative idea about which viruses were present in our Belgian honeybee colonies and whether or not we could associate that with winter loss. So these are 7 viruses. deformed wing and varroa destructor. These are two closely related variants of a virus belonging to the Iflaviridae family. And then 5 viruses, which I have just described in the previous slide. So we developed qPCRs, and then he went back to these 300 original samples that he had from these 300 hives across Belgium and screened them with qPCR for these 7 viruses. So what he found was the following. So first, if we just look at the cumulative data at all the viruses together, there was not really a significant difference between the healthy colonies shown in green and the weak colonies shown in red. However, when we looked at some virus individually, there was actually quite strong association between winter loss and these deformed wing virus and varroa destructor I mentioned before, where we found much higher viral loads of these viruses in the weak colonies shown in red compared to the healthy colonies shown in green. And of course, we have to be very careful. Association doesn't mean causation, but these are certainly intriguing findings that need to be further validated. And also, if we looked at the other viruses, they were in general less prevalent than deformed wing and varroa destructor. But there were also 2 other viruses here, Apis filamentous virus, Apis mellifera filamentous virus, and the orthomyxovirus, which were also more prevalent in the weak colonies compared to the healthy colonies. Interestingly, the Apis tika-like virus, for that one, we found an actually inverse correlation where it seemed that it was more often present in healthy ones compared to weak ones. And whether or not this really has any biological meaning needs to be still investigated, but it certainly is intriguing and would be very interesting if that would really be the case, because that would mean that maybe this viruses would could be used to really spread in bee populations to protect against winter loss. However, this is very preliminary and needs to be further explored. So to summarize this second part of my talk, so we found a plethora of novel honeybee-associated viruses. For the novel orthomyxovirus, we were able to validate with electron microscopy. We found this recombinant Leuven virus that I mentioned, and also this very divergent and potentially interesting Tika-like virus. And then with our qPCR analysis, we identified that the weak colonies had significantly more deformed wing and varroa destructor viral genome copies, and to a lesser extent also Apis mellifera filamentous virus and the orthomyxovirus. And interestingly, there was an association of the strong colonies with more Tika-like virus genome copies. So also for this research, there was also a lot of bacteriophages that we found, and for the sake of time, I will not be able to go into much detail, but this work has been published last year. So if you're interested in the phages of these honeybees, feel free to check out this paper. There's much more interesting content there. And actually the work Wart is doing, there's still several more papers he's preparing where the previous results that I just showed and more will be published. So if you're interested in that, keep an eye on his papers that will come out in the next months. And then finally, the third part of my presentation, this work done by Lynne Beller. She finished her PhD a couple months ago and she now gave birth, so she's now taking care of her own little baby. And what she was interested in was to further investigate how the microbiome of the infants develop over the first year of life. So we all know that our gut microbiome is a complex ecosystem containing multiple organisms. And most research today has focused on bacteria present in the gut. But there are also archaea, which are less common, but there is a whole set of viruses there, which is not always described in very much detail. And also fungi and parasites are present in our gut. So this is a very complex ecosystem, and Lynne was interested to see how this ecosystem developed over time, and especially during the first year of life towards a more adult-like microbiota. So what Lynne did in order to study this was she started a cohort, a longitudinal cohort of Belgian infants, and those were enrolled right after birth. And the very motivated mothers and fathers of these children, collected samples, up 2 to 3 samples per week for the entire first year of the life of that infant. They were initially stored at -20 at home, and then they were transferred to our lab. And with each sample that was collected, the parents also filled in a little questionnaire about the consistency of the sample, what was the dietary pattern at that moment that the child provided the sample. Was there any disease that was noticed? Was there any doctor's visits? Were there any antibiotics used, etc.? Where was the child at the day the sample was taken, etc. So we had a lot of metadata from these infants. So these are the 8 infants that she ended up studying. And on the x-axis, you see the days after birth, so ranging almost, or some up to over 400 days, but most of them ended after the first year of life. And each gray little circle indicates a sample that was collected. So this represents many thousands of samples, unfortunately more than we were able to analyze. So Lynne had to make a selection of samples that she was going to analyze. And this is the selection she made. So each of these green diamonds was a sample she selected. And as you can see, the dark green diamonds, these were all samples that were selected on predefined time points. Towards the second year of life, this was pretty much every month a sample, whereas in the beginning, we selected more frequent time points because we assumed that there would be more changes going on early in life. And then you also have these light green diamonds. These were samples that were selected ad hoc later on based on particular metadata that were available for these infants, as shown here. So what are all these symbols? So actually the black inverted triangles, these were all points where the children received vaccinations. The open triangles here, these were the exact time points that children entered in daycare. And of course, in daycare, they will come into contact with many other infants. And then the inverted brownish triangles were all different events that occurred, such as a gastroenteritis, when children had diarrhea, or they had vomiting, or they had antibiotic treatment, or their teeth started coming through. So a bunch of different points we selected, or events we selected for these infants, and then we selected some time points before and after to see how those events affected the development of the microbiome. So in total, she selected more than 300 samples, around 35 to 40 samples per infant, and those she used in her analysis. So she used these samples to do the virome analysis, as with the method I described before, but she also did 16S to study bacteria on all these samples, and she also did an 18S analysis on all these samples to look for eukaryotes. And initially that was mainly done to find any fungi or parasites, but we could also use that. For example, we found some evidence of plants present there, which were part of the diet of the child. So all of that together allowed her to get a complete, very detailed picture of how the microbiota developed. And again, for the sake of time, I will only have time to go into detail of some parts of the viral work she did. So, as I said, she did the Nativir protocol, she purified all the VLPs, did shotgun sequencing, and she generated about 1 billion reads. And the majority of these were prokaryotic viruses, bacteriophages, which is not a surprise. 12% of the reads were eukaryotic viruses, and also a fraction of what we sequenced were also not viral, as of course the method is not perfect and you can't get rid of all other viruses. But we're quite happy to see that a large percent of the reads that we got were actually viral. So first, I would like to tell you something about the phages. And of course, you get thousands and thousands of phage contexts. But Lynne made a selection on these phages, and she did that based on the fact whether or not the genomes were near complete. So recently, a novel bioinformatics tool came out, CheckV, it's named, which allows to estimate the completeness of a bacteriophage genome. So she selected all the genomes that were predicted to be at least 50% complete. And then she made an additional selection that she took all the phages that were present in at least more than one sample. So if a phage was just present in a single sample of all these infants, it was excluded because it was most likely just something from the diet. So she ended up with 305 near complete phage genomes, which were present at least in more than one sample. And she abbreviated those to the Babel infant gut phages. Babel stands for baby Belgium, and she abbreviated those to the big phages. So she had 305 of these big phages that she further analyzed. And first, if we try to classify them, the majority of these remain unclassified. The classification for phages by the ICTV is still under development, but it's quite tricky. So the majority remained unclassified. And then for the rest, we had some of the usual suspects, the Myoviridae, Poloviridae, Cephuviridae, which are the typical tailed phages, as well as Microviridae. And we also found 2 cross-like phage genomes in 2 of our infants. So the next thing Lynne did was to see how prevalent these different phages were across different infants. And actually the majority of these 305 phages, actually here as you can see, 215 were only present in a single infant. So 70% of all the phages that she found were really infant specific, suggesting that the virome profile, the phage virome profile of different infants is very infant specific. However, there were still almost 8% of these phage that could be found in at least 50% of our infants. And of interest, there were 2 phages that turned out in all 8 of our infants. But when we looked further into it, these were 2 phages that infect lactococcus phages, and they could be statistically linked to the presence of formula feed, so strongly suggesting that this phage might actually be present in this formula feed. So that means that there is not really a set of core phages present in all infants, although some of them were found in 5 or 6 infants, which could also be interesting. So the next thing she did was to see if she could predict the hosts of these bacteriophages. And she used 3 approaches. First, a BLAST-N, just compare it to phages known in databases. And then she looked inside the phage genomes for transfer RNAs, which could be linked to particular bacteria, or for the presence of CRISPR spacers, which could be extracted from bacterial genomes. And doing this approach, she was able to identify or to predict the host in about 80% of her phages. And as you can see here, the predicted hosts are kind of the usual suspects in the human gut. The majority of phage contexts or genomes could be linked to firmicutes and to a lesser extent also to actinobacteria, Bacteroidetes, and Protobacteria phyla. I mentioned to you before that Lynne also did 16 S analysis on all these samples. So that allowed us to try to confirm some of these predictions based on correlations of these longitudinal samples. So what this graph, what this figure shows you here, so these actually are on the top 3 panels are bacterial contexts, so bacterial 16S variants that we found. And at the bottom, we have phage contexts and the number of reads mapping per sample to these phage contexts. And what you can actually see here is that for many of these phages, we could find very nice correlations between the presence of these phage reads as well as the presence of particular host bacteria using 16S. So these are 3 examples where you see very nice correlations between the presence of bacterial strains and the phage context infecting these bacteria. You can also see very often that they appear around the same time. So the first day we detected this bacteria, day 4, that's also the day we detected this phage. In some cases, the phage appears slightly later than the bacterium. So these are 3 examples of bacteria and phages found early in life. There are many more examples. These are a few more from bacteria later in life. Again, seeing very nice co-occurrence patterns of particular bacteria and their phage host. And actually, these findings further confirm a previous observation that was made in another study where the author suggested that when infants are born, their guts are colonized by bacteria. And actually, these bacteria have integrated prophages, which are reactivated and then also start, well, to populate the infant gut. And the fact that we find so many bacteria and such nice correlation with phages kind of confirms that very likely many of these phages that we find here are actually induced from the bacteria that initially colonize the gut of these infants. So if we then take a closer look at the eukaryotic viruses, Lynne had a set of viruses here, which he called the DAMIVs, which stands for disease-associated mammalian infecting viruses. So these are your usual suspects that cause gastroenteritis in infants. And then there was also a group of small circular single-stranded DNA viruses that were mainly the Annelloviridae, and I will come back to those also shortly later. And then we also had some plant-infecting viruses and fungal infecting viruses. But as you can see here, the majority of the eukaryotic virus reads belong to these DAMIVs, so these viruses usually infecting or causing gastroenteritis in infants, and that's also shown here. We have the typical viral families. We expect Reoviridae, which harbors rotaviruses, Picornaviridae, which harbor many viruses, including enteroviruses. We have parvovirus, we have caliciviruses, mainly containing norovirus and sapoviruses, and then astroviruses and adenoviruses, all viruses which have for which it has been established that they can cause gastroenteritis in humans. The small circular virus made up about 7%, and then the plant and the fungi infecting viruses really were a very small minority. So if you look longitudinally at these eukaryotic viruses, in contrast to kind of the phages, we see really that for the eukaryotic viruses, virus really seem to come and go. So this represents probably like acute infections, where the children have an acute infection of a virus that comes and goes in a relatively short time span. As you can see, a peak of a virus, and then it goes away again. And you see that multiple times during the first year of life. These are just two examples, but we saw the same pattern for all the 8 infants with more acute-like infections. So there's no stable colonization of eukaryotic viruses, more acute-like infections. So what Lynne further wanted to do is to check about the frequency and the accumulation of such eukaryotic viruses across the first year of life of an infant. So what you see on the right-hand side here, each line represents an infant, and it also shows you how many different unique eukaryotic viruses we found over time in these infants. So you see that these infants acquired these eukaryotic virus infections over time, with an average about 16 eukaryotic viruses per infant that we could identify in the first year of life. What was also interesting is the following. So you can also see here these black crosses inside these graphs, and these were all time points where the parents of the infants reported some type of disease, some type of enteric disease, either diarrhea or vomiting. So that implies that actually in the vast majority of these infections with eukaryotic viruses, that there didn't seem to be any clinical signs. So that the majority, and actually she calculated was about 86% of these infections were not associated with any enteric signs of vomiting or diarrhea, suggesting that the majority of these eukaryotic virus infections go unnoticed, go asymptomatic in these infants. You can also see probably that in the beginning, it's relatively slow accumulation, and then actually a bit later on, toward the second year of life, it seems to be increasing, that there seems to be an increasing number of eukaryotic viruses that occur. And Lynne did a statistical analysis to see whether or not there could be a correlation between this increase and a number of the metadata we had. And actually what she found was that the entrance into daycare was strongly correlated with an increase in the rate of eukaryotic virus infections. Other things like start of or ending of breastfeeding, or start of formula feeding, starting of fruits, starting of eating vegetables, or just a half a year of age did not correlate, but there was a strong correlation between entrance of daycare. And of course, this makes sense, since then the children meet other children and they happily exchange all the viruses they have. And to quantify this, she calculated that before the entrance in daycare, these children experienced roughly every 48 days a new eukaryotic virus infection, whereas after daycare, this increased to a new infection every 18.5 days. So that was quite interesting, but not all that unexpected. So everything, these numbers I've shown you now, they are excluding the anellovirus. As I mentioned before, that these anellovirus, these viruses with a small circular DNA virome, were also identified. And actually, if you look at the level of the contig, so the number of contigs, then about 80% of all the viral contigs we identified belong to this family of anelloviruses. If we look at the read level, this was only 7.5%, suggesting that there is a large number, a large diversity of these anelloviruses present in these infants, but that they're present at quite low concentrations. And at this point, anelloviruses, there's quite little known about them. We know that they're present pretty much everywhere in our body. They have been found in stool samples, in respiratory samples, in blood samples, etc. They're there, but we don't really know what they are doing, and they're not associated with any type of disease or other condition. We actually found these anellovirus in all the infants we studied. Actually, in pretty much half of all the samples that we analyzed, we could find them. They were usually present for prolonged periods of time. The maximum that we found was that one contig was present for over a year in one of the infants, for which we had data slightly longer than a year. On average, we found those contigs back for 25 days. And also on average, there were about 112 unique anellovirus contigs per infant. So that's a huge amount, but we know very little about these viruses. So that's certainly a very interesting target for further research. So to wrap up this part, the bacteriophages dominate the infant gut, which is not surprising. And we see a gradual colonization of these phages, which strongly correlated with their bacterial host. And that this phage population is quite infant specific, and there is very little that is shared across different infants. Eukaryotic virus infections were most often asymptomatic, and we see a strong correlation with an increase of these eukaryotic virus infections after the children enter daycare. And these anelloviruses are very diverse but low abundant, and at this point we really have no idea what they're doing. So with that, I want to thank particularly the 3 PhD students that did the work I just presented, as well as the rest of our lab, and the fundings agencies, and then also, of course, you for your attention. And I would be happy to try to answer any questions you may have.
Jelle, thank you very much for the nice talk. And I would like to open now the floor for discussion. So please, if some of you who are still linked would like to place some question, unmute your microphone and do so. Would like to ask the technical support if we can see the people who are connected.
Hello, could I ask a question?
Yes, of course. Go ahead.
Hello, I would like to go back to the slides about the virus load in mosquitoes, where you quantified the viral loads. If I understand well the graphs, when you go there.
This one?
This one. Is it that on the x-axis, you have the individual samples, and on the epsilon, you have the loads of viral copies? Yes, that's right. So why does it often have the shape of sort of either very low loads or very high loads? Yeah, it's like exponential phase and then a plateau, like a PCR reaction.
Yes, yes, that's a very good observation. We've also noticed that, but we don't really have a very clear explanation for that. One of the things we could hypothesize that it has to do something with the immune system, that in some of these mosquitoes, you know, mosquitoes usually use the RNAi as one of their main mechanisms to defend. So it could be that there is something with the level of your immune system, or it could potentially be this pure speculation that they might have another related virus seen before, resulting in an upregulated RNAi, which might suppress this, some of these viruses to better than others, but it's pure speculation. So we don't really know why that is, but it is a curious observation that there often seem to be kind of two levels of high and low viral loads of these viruses in these mosquitoes, but we don't really have a clear explanation.
And because you have also the bee samples, can you see something similar? I don't think it was in your slides. I'm just asking because you have the data.
We didn't, well, it probably tried to give a little bit of an idea. We didn't look at it in detail as we did for the other ones. If you would look here, it seems, if you look at the dots, that it's more gradual over different viral loads and less clearly like a bimodal pattern.
Mm-hmm. Yeah. So maybe different immune reactions, or hard to explain.
Yeah, so it could also be, of course, these infections, for example, in bees with these different viruses. It could also be more some transient infection. And then, of course, if you just collect your bee early on in an infection or later on in an infection, that, of course, could influence the viral load you find. If you would have taken a bee now or a few days later, you could find a different viral load for a particular virus in these different bees. So that's probably also playing a role here, just the moment in the infection cycle that you just captured the bee.
Mm-hmm. Okay, thank you very much.
You're welcome.
Okay, is there another question? Okay, that's my colleague here in the hall, so I will invite her to place the question.
Hi, Jelle, Martina. I have two questions. One question is about the Lake Sinai recombinant. So do you think that the recombination is just an accidental process, or it is just the way how the virus may escape the immune systems or something like this?
Yeah, that's a good question. We don't really know. It's the first time we detected it. I think I need to check again that we just found it in one particular sample. So whether or not this is truly a honeybee virus, because we only found it in one sample, it might have been more. It might have been an interspecies transmission from another insect or from a plant. At this point, we really don't know. So as I said, we verified that it's really truly, it's not an artificially generated recombinant. But we also have no idea, is that something that might have happened recent, or is that something that might have happened a very long time ago? That's something we cannot really deduce from the data we have. But this is not something unique that there's other examples in literature where you see recombinations of different viruses resulting in a novel virus. But at this point, well, what you see here is pretty much what we know. So it's hard to say something more about how it emerged or how relevant it is for bees, if it's really actually a bee virus or not.
Okay, thank you. And my second question is about the anelloviruses. I would like to ask you if you correlated the viral load of these anelloviruses to some diseases or vaccination or something which may influence the children's immune systems.
Yeah, so what Lynne also tried to correlate these anelloviruses to the metadata she had, but there was nothing that came out there. But there is some evidence in literature suggesting that these anelloviruses might have some role in immune regulation, because there's, for example, some data where they see that, for example, people that are immune suppressed, either because of a disease or because they need to have a transplant or something, that we see that the levels of these anelloviruses really go up, as suggesting that there is probably some kind of equilibrium between the host immune system and these anelloviruses. And that we assume that they have evolved for probably millions of years together, so they reach some equilibrium stadium. But of course, when the immune system is suppressed for whatever reason, then we see an increase in these anellovirus concentrations. So, but again, the fact that we assume that they probably evolved for a very long time with their hosts probably also makes them non-pathogenic, that they've kind of found some equilibrium. Both happy, the host doesn't have any downsides, and the virus can happily replicate. Although at this point, we have no idea what they actually infect. Do they infect eukaryotic cells? If yes, which ones? So again, there is still a lot we don't know about these anelloviruses.
Yeah, yeah, yeah. Okay, thank you very much.
You're welcome.
All right, so are there any other questions from the audience?
Maybe one question.
Okay, go ahead.
Can you hear me?
Yes.
May any reads from next generation originate from integrated virus into genome?
Yes, so normally the native protocol is designed to purify viral-like particles from your biological sample. And we try to get rid of host cells, for example, bacterial cells or human cells. We try to get rid of those. So in theory, we get rid of viruses that are integrated in their host genome. So we have an enrichment of viruses that are really particles. However, of course, as we have seen, this viral purification is not perfect. So we always get some reads of the host that get through either bacterial or a host of the mosquito or humans. So theoretically, in those reads, you should be able to identify any integrated viral genomes, but those are actually selected against by the procedure. So those we will, if you're really interested in those viruses, then probably the protocol we use is not the way to go.
Thank you.
So are there other questions from the audience?
I have one. I would like if you when you show the correlation between certain viruses in honeybee colonies. In fact, if you see really such a correlation, what is a biological interpretation? I mean, to which extent they really contribute? Yes, to which extent they really contribute to the death of the colony, or to which extent they are just opportunistic, and because of some other primary weakness of the colony, they just come up.
Yes, exactly. That's a very valid point. And I also mentioned that this is an association. And of course, we can't jump to conclusions that this is the cause. So at this point, in the field, that is still, we don't really know. And probably it's multifactorial. So probably it's not just one particular cause that can be identified. So it's very possible that some other factors influence the immune system of the honeybee, maybe suppress it, and that will allow some of these viruses to grow to higher titers, and maybe on their turn also further have a negative impact on the bees, because it's easier for them to replicate. So exactly the exact factors that cause this winter loss, that's not disentangled yet, but it's very likely that it is multifactorial, including probably Varroa destructor. This mite is probably also playing an important role. It might weaken the honeybee just by feeding on them. It might be an important factor for virus. So more viruses get into these bees. There might be environmental factors. So it's probably a combination of many factors that cause these bees to not survive winter. And in different seasons or different places, different factors could be the main one causing the winter loss. But there's not really one factor that has been that we have been able to pinpoint.
Yeah, thank you.
Okay, if I may, one question which is connected to that, where you know that in our mutual project we also screened for parasites, actually, in order to correlate the presence of the viruses with these parasites and the eventual diseases. Do you have data for your samples about the presence of the parasites and
actually,
maybe it's even some bacteria which are causing diseases?
Mm-hmm. So well, there are two parts of an answer. So the samples we used were from the Epilo-bee study that I mentioned, and in that study they have looked also at parasites specifically, but but most of these results have not yet been published. They should have been, but they are not yet. And I don't remember their results by heart. But I don't, if there would be something strong jumping out there, I'm sure I would have heard about it. So I don't think, again, there's a very strong indication there. And with respect to the bacteria, the part I didn't show about the bacteriophages, I think Wart also uses bacteriophages to predict their hosts. And I don't recall that there were any strong links with particular known bacterial pathogens to honeybees. So again, nothing really that jumped out right away. But again, as I said, multiple of these organisms could play a role in the overall winter loss disease. Okay, thank you very much. And one more question from the audience before we finish, if there is some. Okay, I don't hear anyone. So yeah, thank you very much again for spending the time with us and your sharing the data, which are very interesting. I suppose that we will see many papers coming from your lab, and I hope that we will also finish our mutual project publishing the data. So have a good day, all of you have a good day, and thank you for connecting and listening, and we will inform you about other lecture. And also there is a recording from this lecture, so it will be available on the BioSef site, the link for future. Yeah, so thank you very much for the invitation, and I hope on a future occasion I will be able to come physically there, which was now not possible. Yeah, let's hope that can work out in the future. Yeah, well, we hope so too. So have a good day. Have a good day, all of you. Yes, bye-bye. Bye-bye.